In the middle of the 19th century, an Augustinian monk named Gregor Mendel took up the question of biological inheritance with a series of experiments. Mendel had a naturally inquisitive mind and a profound love of nature. His scientific interests ranged from research on plants to meteorology and the theories of evolution. Working at a monastery in what is now the Czech Republic, Mendel started by crossbreeding different strains of garden peas, then observing the characteristics of their offspring. Why choose peas? He did it, he said, for the fun of the thing. Mendel noticed that when he crossed a round pea seed with a wrinkled one, the offspring were round, not a mix of the two characteristics as he had expected. Yet when he bred the round pea offspring, that's where the mix appeared, and the second generation had both round and wrinkled seeds. He continued to experiment, trying to understand what kind of biological mechanism would cause certain characteristics to disappear in the first generation, only to reappear in the second. Then one day, Mendel counted the number of peas in the second generation that had the wrinkled characteristic. Exactly 1/4 of the peas. For Mendel, here was the breakthrough. For the first time, he could demonstrate that the traits of successive generations were inherited in certain numerical ratios. In other words, there were fixed laws of nature that governed heredity. With this insight, Mendel made the first great discovery in the science of genetics. Each inherited characteristic must be decided by a pair of what he called factors. Each parent, he said, contributes one factor for each characteristic. Certain factors are dominant and others recessive, depending on the combination of the factors the offspring inherits. And Mendel's factors are called genes. The term Mendelian trait is used to describe a characteristic caused by a single gene that sometimes reappears in 1/4 of the offspring. That characteristic can be innocuous, such as freckles or the ability to curl your tongue. But it also can lead to serious illnesses like cystic fibrosis or Tay-Sachs disease. Imagine all that from... The story goes that one day, shortly after Morgan started breeding Drosophila for his experiments, a striking mutant appeared in his lab. A fly with distinctive white eyes. He decided to breed it with a female fly that had ordinary red eyes, curious to see what the offspring would look like. 2 weeks later, he had his answer. One by one, the first generation offspring appeared. All of them had red eyes. Thinking that the white eyes might be a hidden characteristic similar to what Mendel had observed, Morgan waited to see what would happen in the second generation. Sure enough, the characteristic skipped a generation. This time, some of the flies had red eyes and some had white eyes. But then Morgan saw something else. All of the flies with white eyes were male. At the time, it was known that the gender of a species is determined by 2 of the rod-shaped structures found in the cell nucleus, the chromosomes. For example, human females have 2 X chromosomes. Human males have 1 X chromosome and 1 Y chromosome. Morgan realized that the gene responsible for the white eyes must somehow be associated with the fact that male flies have only 1 X chromosome. This meant that in the females, the gene responsible for red eyes on one of the X chromosomes might be overshadowing the gene for white eyes on the other. To prove his point, Morgan bred thousands of fruit flies and studied their inheritance. One of the geneticists who studied fruit flies alongside Morgan was George Beadle. Working in Paris in 1935, Beadle detected evidence that the inherited trait of eye color in Drosophila might be the result of genetically based chemical reactions. Beadle pursued his investigations at Stanford University with colleague Edward Tatum. For their experiments, Beadle and Tatum selected another simple organism, Neurospora crassa, bread mold, They chose mold because it was easy to grow with simple nutritional needs: bread, air, and water. Neurospora also had a single set of chromosomes, which allowed the researchers to observe genetic changes easily. Knowing that X-rays damage the chromosomes, Beetle and Tatum irradiated the mold, which caused mutations in the genes of its spores. These mutant genes were unable to produce the nutrients necessary for the mold to grow. However, when they added the nutrients, some of the spores began to germinate. Discovery as well. It happened here in 1952. Biologists Alfred Hershey and Martha Chase were studying bacteriophage, a virus known to infect bacteria. Through a process called transformation, the virus takes over the bacterium's internal machinery and commandeers it to produce more viruses. Bacteriophage consists of two simple components: a protein shell, and inside, a mysterious substance scientists had known about for decades. After using a kitchen blender to separate the bacteria from the empty viral protein shells, Hershey and Chase saw that the radioactive protein was not penetrating the bacterial wall. But inside the bacterium was the radioactive DNA. It was the DNA, not the protein, that was able to direct the host cell to make new viruses. In a 1953 issue of Nature magazine, Watson and Crick revealed their amazing discovery to the world. Their model showed that the DNA molecule was a double helix. The twin strands were composed of pairs of the 4 known bases linked together by hydrogen bonds. And the whole structure corkscrewed like a spiral ladder, which could easily pull apart in order to make copies of itself with the same encoded genetic information. Years before Watson and Crick had defined the structure of DNA, scientists knew that DNA was responsible for making proteins that resided in the cytoplasm. But they were puzzled by how the DNA managed to transfer its genetic information for making proteins through the nucleus wall. The answer to the puzzle came through the collective work of several different scientists. And like many discoveries, it began with an educated guess. The production of protein in a cell is seen here in red. Scientists found that cells with lots of protein production contained lots of this, RNA, a chemical similar to DNA, but with only one strand, not two. This led scientists to wonder if RNA was somehow involved in running the protein factories in the cytoplasm. To find out, once again, researchers turned to the bacteriophage. They detected that soon after the bacteriophage injected its DNA into a bacterium, traces of viral RNA appeared in the host cell, and the production of proteins began to increase. This was the moment of discovery. Here was a special kind of RNA never seen before. As its single strand enters one of the cell's protein factories, it communicates the messages for making new protein. Scientists called it messenger RNA. Today, many scientists believe that messenger RNA and other RNA molecules are descendants of the Earth's most primitive chemicals. As we've just seen, RNA translates instructions from DNA for making proteins. But what was the genetic code, the sequence of instructions that made this process possible? In 1961, molecular biologist Marshall Nirenberg and postdoctoral fellow Heinrich Mattei carried out a series of experiments to see if they could synthesize proteins in the laboratory. They already knew that there are 20 amino acids involved in the production of proteins. In their experiment, Nirenberg and Mattei were working with RNA. Like DNA, RNA is made of 4 chemical bases. In a eureka moment of discovery, They found that when 3 of the bases align in a specific sequence called a triplet, the triplet codes for a specific amino acid. The order of the triplets is the blueprint for the production of proteins. A peek at the secret vocabulary encrypted in each DNA molecule, enabling it to instruct the synthesis of proteins. Over the next 5 years, Nierenberg successfully deciphered the 64 triplets that comprise the entire vocabulary of DNA. The genetic code had been broken. And in 1968, the discovery earned Marshall Nierenberg a share of the Nobel Prize for Physiology and the breakthroughs that led to some of the discoveries we've seen so far might never have happened if not for this familiar organism, bacteriophage. And our next discovery depended on it as well. For decades, it was believed that bacteria were completely vulnerable to invading bacteriophage. But in the 1950s, researchers found a wrinkle in that view. Certain types of bacteria were, in fact, resistant to being infected by a bacteriophage. How is that possible? The first answer was provided in 1962 by microbiologist Werner Arber. He found that some bacteria had enzymes that fought back against the virus by cutting its viral DNA into pieces. This restricted the virus from taking over the bacteria. So they were called restriction enzymes. But how exactly did they work? Among those hoping to unlock that secret was microbiologist Hamilton Smith. Working in his lab in 1972, Smith was growing bacteria and bacteriophage together when he noticed that the DNA of the virus was breaking down. Smith acted quickly. He purified the restriction enzyme. Then he identified the exact place where the restriction enzyme had severed the DNA. Then came the moment of discovery. Smith found that the enzyme repeatedly cut the viral DNA in the same place. He had discovered the first site-specific restriction enzyme. London, 1985. A boy arrives by plane from the African nation of Ghana. The purpose of his visit? To be reunited with his mother. But custom inspectors are suspicious. The boy's passport appears to be a forgery, and there's no proof that the woman he's meeting is really his mother. The British government decides to deport the boy. Desperate, the woman turns to a detective for help. His name? Alec Jeffreys. Not a real detective, but a geneticist at Leicester University. In 1984, Jeffreys found something never seen before. It was a DNA variant formed by short identical sequences of DNA that were repeated over and over. Jeffreys called these repeated sequences mini-satellite DNA. Then came the breakthrough discovery.
What we're detecting are lots of bits of DNA, and in the humans at least, are clearly variable, very variable. And in fact, what we got was our first very, very murky DNA fingerprint. And at that point, the penny dropped.
Jeffreys' discovery made history when he was asked to help solve the mystery of the boy from Ghana. For the first time, DNA fingerprinting was put to the test. Jeffreys compared the DNA of the boy with the DNA of his supposed mother. The results showed a striking similarity in the mini-satellite DNA, proving beyond a doubt that the boy was her son.
So I was there when she was told, we've got the DNA evidence, it's been accepted, your boy's coming back, he's permanently with you. And it was the look in that lady's eyes was magic. And this was really, the first time DNA had ever done this. It was the first time that DNA, all this modern arcane science of molecular genetics, had actually been used to go out in a non-clinical context and actually directly help someone.
By June 2000, the two teams were ready to make history.
Today, marks a historic point in the 100,000-year record of humanity. We are announcing today, for the first time, our species can read the chemical letters of its